Ayun. So, good afternoon ulit, class. Um, Welcome again to our subject, Molecular Biology and Diagnostics. And last time, wait lang, naririnig niyo ba ako? Baka di niyo pala ako naririnig. Pa-confirm kung naririnig niyo ako.
Naririnig po.
Okay, thank you. Thank you. Okay. Naririnig niyo naman ako. Okay. So, last time, we discussed about DNA structure, hereditary information, and DNA replication. So, halos review lang yun lahat sa inyo, no? Dahil sa cytogenetics class nyo. And, yung gusto ko sana i-emphasize dun sa DNA replication ay yung PCR. Kaso, sabi nga ni Sir Isan, yun sana yung gagawin natin ngayong hapon, magpi-PCR tayo. Kaso, inorient ako ni Sir Isan na tapusin muna lahat ng lectures, tapos sa May na lang yung lahat ng laboratory. So, i-postpone natin yung PCR. Pero during our lab discussion on PCR, tuturuan ko kayo kung paano mag-PCR, like in-depth discussion and yung step-by-step na methods para mag-PCR. So, kapag mag-PCR na kayo sa medtech career nyo, alam nyo na kung paano. Okay? So, ay wait lang. Okay, okay. So, let's start our discussion for this afternoon. So, again, we discussed about the DNA replication. Ito, dito na tayo. Wala palang pointer. Dito na tayo sa DNA replication. Tapos na natin. And also DNA structure and hereditary information. This afternoon, ang i-discuss natin ay about DNA repair and recombination. So, di pa rin tayo umaalis dito sa DNA. Pero hayaan nyo, next meetings, lilipat na tayo sa transcription and translation. So, before we start our meeting, wait lang. Before we start our meeting, di lumalabas. Sad. Ayan. Ito yung slide na gusto kong ipakita. We all go through things differently, right? So, how deep is the mud depends who you ask. So, kaya ko lahat ng activity na binigay ko sa inyo, sabi ko, 'di ba, by the end of the sem na, or ang deadline ng lahat ng 'yon ay end ng sem na to or last week ng sem na to. Pero in-encourage ko kayo na ipasa pa rin sila ng on time para may sense kayo ng progress and para ma-review nyo yung lahat ng inaaral natin, especially yung quizzes. So kapag on time nyo sagutin rin yung quizzes, on time din kayo makakapag-review. Okay. So, dahil nga iba yung panahon natin and kailangan din natin mag-adjust sa mga sitwasyon dahil alam ko yung iba sa inyo merong mga sitwasyon na medyo may struggles or may challenges, pero yun, kaya nyo yan, guys. Graduate din kayo ng medtech at magiging RMP. So, start na natin yung lecture. Yun lang. So today, our discussion is on DNA repair and recombination. So we are going to discuss basically the damage in how the DNA is damaged and how it is repaired, and we are going to learn the various DNA recombination mechanisms, and we are going to study the diseases related to the dysfunction of the repair mechanism. So ito si Darion Nguyen, kilala niyo ba siya? Pero hindi nakikita yung video. Sad. Hindi nagpi-play yung video. So, TikTokerist siya. TikTokerist siya ng mga laboratory techniques and scientific stuff. So, pwede niyo siya i-follow sa Lab Shenanigans. And meron siyang TikTok tungkol sa eukaryotic DNA polymerase. It makes errors every 10,000 to 100 nucleotides. Pero dahil nga merong proofreading mechanism ang DNA nucleotide, mas bumababa yung mutation rate. So, na-discuss natin yun last time, right? So, yung mutation rate lang sa humans, again, one mutation per 10 billion nucleotide base pairs per cell generation, right? So, ayun. Um, and we discussed DNA polymerase proofreading, which basically ito yung simplification niya. So, the polymerase, it adds an incorrect nucleotide to the new DNA strand, right? But remember that the DNA polymerase has two sites, the polymerizing site and the editing site. And the the polymerizing site adds the new nucleotide. Now, your editing site, it detects the bases which are mispaired. And once it detects the bases which are mispaired, the DNA polymerase, it has an exonuclease activity to remove the incorrect nucleotide. So, basically, yung DNA polymerase natin, meron siyang sariling proofreading mechanism. And again, it allows us to have a low mutation rate. So, now, DNA damage and repair, kailangan natin itong kailangan natin itong ma sorry, may mental block po. Kailangan natin itong ma-avoid. So, how do we avoid DNA damage and repair? So, we are going to discuss this today. And itong picture lang na to, picture siya ng DNA repair na nangyayari na parang merong researchers from researchers from Harvard School of Medicine, they were curious on how the broken DNA ends get reconnected with each other. So they labeled the ends of the DNA and they with fluorescent markers, and they observed that once these DNA ends connect, they change in color. So yun lang yung tinitingnan nila dito. Parang gusto lang nila makita on how uh, these DNA damages are repaired. So they label the ends of the DNA with fluorescent markers. And without DNA repair, spontaneous DNA damage would rapidly change the DNA sequences. And even under normal cell conditions, the DNA is susceptible to damage. And these damages they lead to mutations. And if they are not repaired, they will cause certain diseases. And here are the spontaneous DNA damages that happens to our DNA. So first is oxidative damage. Next is hydrolytic attack, and last is methylation. And this figure only shows the sites on each nucleotide where damage occurs. So ano lang to, parang representation of the sites on where in the nucleotides the damage occurs. So the red arrows, they indicate oxidative damage, while the blue arrows, they indicate the hydrolytic attack, and the green indicates the methylation. And in methylation, as you can see in this figure, um, for example, you have a nucleotide with guanine as base pair. There are harmful chemicals which act upon the nucleotide, and these um, results to the addition of the methyl group here. So, ito lang naman yung methylation. So, due to harmful chemicals, methyl groups are added to your nucleotide bases. And type of wait lang. Another type of damage is depurination. So, the human cell, it loses about 80,000 purine bases every day because of depurination. And depurination is basically where your glycosyl linkage of purine to deoxyribose hydrolyze. So, ano bang ibig sabihin nito? So, in depurination, natatanggal yung purine mo. And your purines are guanine and adenines, right? So, yung glycosyl linkage daw dito, Remember, this is glycosyl linkage, right? Yung nagli-link sa sugar and yung nucleotide base. Yung glycosyl linkage mo daw, naka-hydrolyze. Meaning, um, yung water, it acts upon the glycosyl linkage, therefore, displacing your base, your nucleotide base. So, tingnan nyo dito, when your glycosyl linkage is hydrolyzed, matatanggal yung purine mo. So, nawala dito yung guanine mo, right? Ang depurination, it happens only on your purines, um, guanine, and adenine. So, in depurination, again, your glycosyl linkage here is hydrolyzed, okay? Thus, removing your purines. And your purines are substituted with a hydroxyl group here. So, yun yung depurination. Okay? So, in depurination, once it happens, there is a mutation, specifically deletion that happens. So, when your base here is depurinated, or your nucleotide here is depurinated, your purine, in this case adenine, is removed. Di ba nawala? Now, during DNA replication, if, di ba ito, strand na to, ginagamit for DNA replication and ito rin na isang strand. When you use this as a template, yung merong mutation, when you use this as a template, merong kang mutation na mangyayari dito. So, yung new DNA mo ngayon, mawawalan siya ng 80 nucleotide. Okay? Na-delete yung 80 nucleotide dito. Because again, na-depurinate yung nucleotide mo dito. Okay? Then, another example of damage is deamination. So, deamination naman is the conversion of nucleotide to a natural product by hydrolysis of the amino group. So, the most common example is cytosine. Okay? So, in deamination, what basically happens is that your amino group is hydrolyzed. So, look here in your nucleotide base. Yung amino group mo dito, ito yung amino group, right? Na-hydrolyze siya. And once it is hydrolyzed, it is removed. So, mababago na yung nakalagay mo dito. Oxygen atom yung nakalagay mo dito. Therefore, this whole nucleotide will change into uracil. Yung nucleotide mo ngayon, yung naka-attach na sa kanya ay uracil. Okay. Sabihin nyo kung masyado akong mabilis. So, ito. Here in this figure, you will see the deamination in your purines, adenine and guanine. So, remember yung structure ng nucleotide bases mo, right? So, yung adenine mo, merong amino group, okay? And this amino group is hydrolyzed, therefore displacing the amino group. Then, magkakaroon ka ngayon ng a natural product which is called hypoxanthine. Yun yung pangalan ng molecule na to. Kapag na-deaminate mo yung adenine, magkakaroon ka ng hypoxanthine. Okay? Meanwhile, in guanine, this is the amino group, right? So, yung amino group mo, it is hydrolyzed, therefore displacing the amino group. And mababago na yung naka-attach dito, oxygen na. And yung magiging product ay xanthine. Okay? So, when you deaminate guanine, you will produce xanthine. Meanwhile, when you deaminate adenine, you will produce hypoxanthine. Okay? And for your pyrimidines naman, here as you can see, your cytosine, here is an amino group, right? So when you hydrolyze this amino group, removing the amino group, you will form uracil. Okay? But then here, look at thymine. Thymine, wala daw deamination. Bakit kaya? So yung thymine, Walang deamination dahil wala siyang amino group, right? Tingnan nyo. Amino group, merong amino group yung cytosine, pero yung thymine, wala. So, hindi mo madi-deaminate yung thymine. So, yun lang yung specific deaminations in your, each of your nucleotide bases. Okay. So, deamination, um, again, it creates mutation. So, for deamination, yung magiging mutation mo ay substitution. So, in deamination here, when you deaminate again cytosine, it will become uracil, right? Balikan natin. So, ito cytosine, magiging siyang uracil if it is deaminated. So, once the cytosine is deaminated into uracil, there is a change in the template strand. And when that template strand is used as a template for DNA synthesis, magkakaroon ka ng bago dito na nucleotide base. So, imbes na um, G, 'di ba? G yung tama, guanine yung tama na nucleotide base. Yung nagkaroon ay adenine. So this is called substitution mutation. So in comparison to depurination, yung naging mutation ay deletion. And in deamination, yung naging mutation ay substitution. Okay. And another damage is called the pyrimidine dimers. So in pyrimidine dimers, what happens is that yung UV light mo, it um, affects your nucleotide bases. Therefore, yung pyrimidine, specifically your pyrimidine nucleotide bases, so yung pyrimidine nucleotide bases mo, they form covalent bonds. And these covalent bonds, once they are formed, it forms a dimer. So yung pinaka common na dimer ay yung thymine dimer. And this forms a kink in your DNA strand. So, as you can see in the figure, yung UV light from the sun, it causes these thymine bases to create a covalent bond between them. And again, this forms the thymine dimer and in turn forms a kink in the DNA double helix. Okay. And another damage is that there are breaks in your DNA caused by um, X-rays or gamma rays, so or other environmental factors. So one is your single strand break and another is your double strand break. Okay. So those are your damages in your DNA. So yung first, 'di ba, balikan natin. Yung first ay spontaneous DNA damage, oxidative damage, hydrolytic, and methylation. And we've also looked at depurination and deamination. And also, the formation of your pyrimidine dimers.
Ma'am,
Yes po.
yung thymine dimers po, edi ma'am nagko-cause po siya ng parang ng disruption sa transcription?
May, magkakaroon siya ng problema sa transcription if hindi siya makorek. So, yung i-discuss natin next, yung ways na para matanggal itong thymine dimers mo and makorek para pag transcription na, mawawala na siya. So, pero merong cases na nakakalusot tong thymine dimers mo and specifically in the disease Seroderma pigmentosum, which we will discuss later in this lecture. Okay. So, ito munang, pinapakita ko muna sa inyo yung types of DNA damage. And so, if these DNA damages are not repaired and they accumulate, then diseases will occur. Parang wala na, finish na. So, tama si Leila na kapag itong thymine dimers na to, parang ma-carry on pa in transcription, magkakaroon tayo ng diseases. So, that is why we need to repair them. And luckily, our cells, they have the ability to repair DNA damages. So, here are, so, DNA damages are easily repaired because the DNA double helix is parang sobrang ideal niya. Why? Because the DNA double helix, again, is a complementary strand. So you can use either of the strands to correct a mutated strand. So if this strand is mutated, you can use this strand for as a template for the mutation or para makorek yung mutation. So meron siyang advantage. And the first type of DNA repair is translation polymerase. So the replicative polymerase is switched out for specialized DNA polymerases and replicates past the damage. And yung DNA polymerase na ginagamit dito sa eukaryotes ay yung DNA polymerase ETA. So in this figure, as you can see, meron ditong DNA damage, right? Ito yung DNA polymerase mo. So ngayon, na-detect na merong DNA damage dito. Therefore, yung DNA polymerase mo, marirelease and mapapalitan siya ng translation DNA polymerase. And this translation DNA polymerase is in the form of DNA polymerase ETA in eukaryotes. So this DNA translation DNA polymerase, what it does is um, it continues to replicate your DNA. So, yung DNA damage dito usually ay thymine dimers. So, ito yung tinatanong ni Lela kung paano siya makokorek. So, um, yung translation DNA polymerase mo, it has the ability to remove your thymine dimers. And, actually, no. Sorry. Correction. Your translation DNA polymerase, it has the ability to put nucleotide sequences in the growing strand kahit na merong thymine dimer yung template strand mo. So, for example, yung DNA damage mo dito, may thymine dimer. So, T, right? Thymine dimer. Now, yung DNA polymerase mo, translation DNA polymerase mo, kahit na may thymine dimer dyan, mag-a-add pa rin siya ng adenine. Okay? So, even though there is a mutation or a damage here, it will add your specific base or your correct base pairs or your correct bases in the form of adenine. So, nagets nyo ba yung sinabi ko or magulo? Sorry, sabog-sabog ako ngayong araw. Nagets nyo ba? Okay. Proceed na lang tayo. So, there are again breaks in the DNA. And the first one is the single strand break. And let's look at the ways on how the single strand break is repaired. So first is mismatch repair. Next is base excision repair. And lastly is nucleotide excision repair. Isa-isahin natin. So mismatch repair, it is, wait lang. Mismatch repair is the backup repair during DNA replication, as we've studied in our previous lecture. And a genetic defect such as the Lynch syndrome is attributed to the dysfunction of the mismatch repair. The second one is base excision repair. And this is caused by carcinogenic exposure such as tobacco smoking and or diesel exhaust. Or yung dysfunction ng base excision repair ay attributed to carcinogenic exposure. And lastly is yung nucleotide excision repair mo, which is it is the most commonly used repair for DNA damages which are bulky. So for example, in your thymine dimers, nucleotide excision repair yung ginagamit din. And these are attributed to some damages caused by ultraviolet light and yung disease na attributed din sa dysfunction ng nucleotide excision repair ay xeroderma pigmentosum. Okay. So let's take a look at mismatch repair. So in mismatch repair, merong kang nade-detect na mismatch in your newly synthesized strand. So as you can see here, thymine yung complementary base pair mo. But the added nucleotide strand is guanine. So a mismatch is detected in the newly synthesized DNA. And then the new DNA strands is cut and mispaired nucleotides and its neighbors are removed. So merong whole section dito ng nucleotides na na-remove. And then the missing patch is replaced with correct nucleotides by DNA polymerase. So once this is removed, nilalagyan dito ng DNA polymerase nung tamang nucleotide bases. And then lastly, your DNA ligase fills the gap in the DNA backbone. And ito lang yung parang simplification ng mismatch repair. But wait, there's more. Sa mismatch repair, you have your um, proteins. So this is called the mut S and mut L complex, which they detect your mismatch in the DNA. So, yung mut S and mut L mo, these proteins detect the mismatch in your DNA. And then, yung mut H mo, it um, detects a certain sequence in your DNA called GATC. And once it detects the GATC, your certain sequence, it binds to the complex. And once it binds to the complex, it bends your DNA. So, kapag na-bend na to, um, yung mut H mo, it makes a cut here. It makes a cut here until here in the mismatched base pair. So, once it makes the cut, your DNA polymerase can now add the nucleotide base pairs which are correct. So, sa way na 'to, parang ang daming section niya na tinanggal para lang sa isang mismatch. And here, the mismatch has been repaired correctly. So, yun yung mismatch repair up close. Meron kang, again, proteins, mut S, mut L, and mut H. And later on, we will see that the mutation in this mismatch repair cause certain diseases. So the next type is base excision repair. So for example, you have a deamination which converts your cytosine to uracil, right? Remember deamination. So uracil yung nandito imbes na cytosine. And then yung uracil mo, it is detected and removed, leaving a baseless nucleotide. Tinanggal dito yung uracil mo. And then yung sugar phosphate backbone mo din, tinanggal din. So, therefore, magkakaroon ka dito ng small gap in your DNA. And once the nucleotide base is excised or removed, the hole is filled with the right base by the DNA polymerase and the gap is filled by ligase. So, sobrang simple lang ng base excision repair. So, yun yung simplification of base excision repair. But there are actually enzymes which are responsible in your base excision repair. So, the first one is DNA glycosylases. These enzymes, they recognize and remove the unnatural nucleotide. So, in this case, um, again, a deaminated cytosine which became uracil. So, DNA glycosylases mo is an enzyme na-detect niya na merong mali dito. Okay? So, once the uracil is detected, your DNA glycosylase removes your uracil. Okay? So, once that is removed, the DNA helix is missing a base. And yung missing base dito, nadedetect siya ng AP endonuclease. And it removes the sugar phosphate backbone. So in this step, your AP endonuclease and also a phosphodiesterase, these are enzymes which removes your sugar phosphate backbone here. Okay? Therefore producing a gap in your DNA. And this gap is again filled with new nucleotide and the nick is sealed by the DNA ligase. Okay? So, bakit flipping out yung sa DNA glycosylases? Kasi, actually, flip out niya tong nucleotide bases dito, parang pinapalabas niya. Yun lang naman yung meaning nun. Flip out niya. And AP endonuclease missing tooth kasi, again, dito, AP endonuclease mo, it removes your phosphodiester, or your sugar phosphate backbone, therefore magkakaroon ka ng missing tooth. Okay? Yun lang naman yung parang keywords para maalala nyo sila. Okay. The next type of repair is nucleotide excision repair. So this repair is used for bulky lesions. So bulky lesions, such as covalent reactions of DNA bases with large hydrocarbons by carcinogens caused by carcinogens such as benzopyrene found in tobacco smoke, coal tar, and diesel exhaust, and pyrimidine dimers caused by sunlight. So, basically, yung sinasabi dito, yung nucleotide excision repair, para siya sa bulky lesions caused by covalent reactions of carcinogens, and also in the pyrimidine dimers caused by sunlight. So, as you've seen in our previous slide, yung pyrimidine dimers, right? It is an example of a bulky lesion. So in nucleotide excision repair, for example, here you have a thymine dimer caused by UV radiation. Um, Madedetect siya, and then the surrounding DNA is opened to form a bubble. And these cut by enzymes, or the enzymes cut the damaged region out of the bubble. And then your DNA polymerase again adds new nucleotide sequences which are correct. Mhm. So ito yung enzymes responsible or enzymes responsible in nucleotide excision repair. So first is your excision nuclease. And your excision nuclease, it basically detects the error. For example, here, meron ditong CT dimer. Type rin siya ng pyrimidine dimer because cytosine and thymine are both pyrimidine. And then, your excision nuclease, um, it marks the section of the DNA to be removed. And your DNA helicase, it removes the strand of the DNA which has the pyrimidine dimers. And once that is removed, your DNA polymerase now adds your correct DNA bases. And again, lastly, your DNA ligase, it connects the gaps between those uh, strands. Okay. So, yun yung mga DNA repair in your single strand break. So, how about in your double strand breaks? So, in your double strand breaks, there are two types, non-homologous end joining and homologous recombination. So, in non-homologous end joining, you've seen this image dun sa first part ng slides natin. Yung scientist in Harvard School of Medicine, what they wanted to observe is how yung non-homologous ends mo join. So they use this technique called FRET, fluorescence resonance energy transfer microscopy, which basically once your DNA connects, magbabago ng kulay. So nagiging red. So when your DNA connects, or joins when your DNA ends joins, magbabago ng kulay yung fluorescence. So, in this way, um, makikita or ma-observe mo yung in-between phases ng DNA, ng non-homologous end joining. So, kasi dati, ang alam lang nila, magbe-break yung DNA tapos magjo-join na. But, hindi pa nila na-observe kung paano sila or kung paano nagko-connect yung ends na yon. So, kaya nila ginawa tong experiment na to using FRET or fluorescence resonance energy transfer. Okay. So, wait lang. Non-homologous end joining is very simple. It just glues your DNA strands together, and usually with a small mutation at the break site. So, sobrang simple niya. Kaya ito rin tinawag na quick and dirty. Quick and dirty. Kasi parang kinoconnect-connect niya lang yung DNA ng yung ends lang ng DNA. So, yun. Sobrang simple lang ng non-homologous end joining. So, yung end ng DNA na na-break, so double-stranded breaks, right, kinoconnect niya lang. So, next is your homologous recombination. And in homologous recombination, the broken the damaged region is replaced by a recombination using sequences copied from the homolog. So titingnan natin up close. So in homologous recombination, you have your double strand break here, right? And when you make a daughter DNA, dalawa yung magagawa mong DNA double helix, right? So, ito yung isang daughter DNA, tapos ito yung isa pang daughter DNA. Now, yung isa daw na daughter DNA, nagkaroon ng double strand break. So, this needs help, right? So, now, yung nuclease mo, ida-digest yung region um, ng isang daughter DNA na nagkaroon ng double strand break and magkakaroon ng strand exchange doon sa isang daughter DNA sa red. So, biglang papasok to dito. Why? Para makopy niya yung nawala or makopy niya ng maayos yung nawalang section ng DNA. Okay? So, yung repair polymer, once na magkaroon ng strand exchange, yung repair polymerase synthesizes the DNA, represented in green, using undamaged DNA as your template. So, itong DNA strand na to, ginagamit niya tong isang DNA strand as a template. And then, the invading strand is released and broken double helix is reformed. So, since meron ka ng copy dito from this DNA template, i-release mo na siya, and then you can use this as a template to make the broken section in this other one. So, tingnan nyo dito, itong section na to, pwede itong i-copy ng broken section dito. And also, yung section dito, pwede nang i-copy ng broken section dito. And then, DNA synthesis continues using strands from damaged DNA as template. And lastly, magkakaroon ng DNA ligation. So, ito yung animation para hindi nyo i-imagine lang.
Ma'am, yung homologous recombination po, similar po yan sa crossing over?
Yung homologous recombination, term din siya na ginagamit for crossing over in your meiosis ba? Meiosis. Meiosis? So, yung homologous recombination, general term siya for yung pag-crossover ng isang DNA section to another DNA section. So, usually these DNA sections are alleles or they have similar hereditary information. And in crossing over, yung crossing over, papalitan sila para yung genetic information mo from your mom and your dad maghalo. So, yung homologous recombination in that sense ay sa paggawa ng meiosis. But here in DNA repair, there is also a term na homologous recombination, pero sa sense na to, para siya sa DNA repair ng damaged DNA na may double strand break. Nasagot ko ba question mo?
Yes po, ma'am. Thank you po.
Okay. Thank you. Okay, so yan yung homologous recombination sa DNA repair for double strand breaks. Okay, and makita natin na mamaya important siya sa certain disease which is actually breast and ovarian cancer. So, ito. So, homologous recombination is also used in the repair of your broken replication fork. So, sometimes, your replication fork nagkakaroon ng break. So, similarly, homologous recombination is used. So, magkakaroon ka lang naman dito again. Yung nuclease mo, i-degrade yung part which has a nick or has a damage. And then, once your nuclease degrades, meron ka dito ngayon na overhang which goes into the strand na undamaged. And this is termed as strand exchange. And in strand exchange, yung damaged DNA mo, ginagamit niya yung undamaged DNA as a template. So now, once the strand breakage and additional synthesis happens, pabalik or pwede ka na ulit magkaroon ng DNA replication sa part na to and na-solve mo yung problem ng DNA damage dito. So, as you can see here, nagkaroon ka ng resume or nag-continue yung replication fork mo. Okay? Mhm, mhm. So, here now, para ma-appreciate yung inaaral natin, ito yung diseases which are attributed to the dysfunction of your DNA repair. So yung first ay hereditary non-polyposis colon cancer, which is also called your Lynch syndrome. And it is a genetic disorder that causes an increased risk of developing cancer, which is, yung Lynch syndrome, general term siya for genetic disorder that causes an increased risk in developing cancer, but here we are going to focus on hereditary non-polyposis colon cancer or HNPCC. And HNPCC is autosomal dominant and there are mutations in the genes involved in the DNA repair, which are mismatch repair genes or MMR genes. Okay? Wait. So, in hereditary non-polyposis cancer, you diagnose it by using microsatellite instability testing. So what is microsatellite instability testing? Um, Microsatellite instability, it is the condition in which merong hypermutability sa impaired DNA mo. Or the presence of MSI, basically, evidence siya na yung mismatch repair genes mo hindi nagpo-function properly. So, a microsatellite in your genetics class, it is a structure with repeated nucleotides, mostly often seen as GTCA, repeats siya ng GTCA. And researchers, they found out that when you look at your microsatellites, you can actually determine if that person has HNPCC. Why? Because dito sa normal microsatellite replication, tama yung replication ng DNA mo. But in abnormal mismatch repair defect, yung microsatellites mo, mayroong variability in sizes. So, again, your microsatellites are repeats in your DNA. And in the normal cell, when your mismatch repair is working properly, yung microsatellites mo properly rin nare-replicate. Now, if your cell is abnormal and meron kang defect in your mismatch repair genes, meron kang variability in the size of your microsatellites. So, yung variability in your microsatellites, this is called your microsatellite instability. And this is tested through PCR. So, ito na yung parang output ng real-time PCR. So, gusto ko sana i-discuss muna yung PCR sa inyo bago to, pero, well, para maintindihan nyo na lang, sa PCR, in polymerase chain reaction, what we do is that we amplify a certain region of the DNA, right? So here in microsatellite instability testing, what we are going to amplify are the genes for your are the genes with microsatellites or mali, are your microsatellites. Yung i-amplify natin dito are your microsatellites. Okay? Yung repeat. So here in MSIH, microsatellite instability high, focus muna tayo dito sa taas. Ito raw yung samples mo with microsatellite instability. So therefore, yung variability ng microsatellite mataas. And itong FAM, Hex, Texas Red, yan, fluorophores yan na ginagamit sa PCR. And these fluorophores, Sila lang yung parang nagbibigay ng signal if na-detect mo yung gene, yung section ng DNA. So, you use FAM, hex, tetrathread, or other types of fluorophores. So, fluorophores lang to. Okay? And, sabi dito, yung normal tissue daw, yung blue. Ito yung blue. Normal tissue, blue. But in your tumor tissue, itong red, tingnan nyo, hindi siya pareho. Mhm. So, ito yung wave na parang ginagamit for detecting microsatellite instabilities because in your normal tissue, parang kino-compare mo yung microsatellite markers mo in your normal tissue and in your tumor cells. And nalimutan ko pala ipakita. Itong BAT25, NH24, NH21, yang mga na yan, markers lang, microsatellite markers lang yan. So, these microsatellite markers, yan yung ina-amplify mo. And as you can see, in your normal tissue, ito yung itsura ng amplified ng microsatellite marker. But hindi siya nagtutugma sa tumor tissue. So therefore, meron kang instability. And as compared to MSS or stable microsatellite, as you can see, nagtutugma yung microsatellites mo. Okay? Malinaw ba? Or naguluhan kayo? Malinaw ba? Or naguluhan kayo? Paano ko ba to explain ng mas malinaw? Get nyo ba yung, yung basic mechanism behind PCR? Mm-hmm. So, again, in PCR, mag-amplify tayo ng genes, right? And in this case, yung i-amplify natin ay yung microsatellites. And ito lang yung parang names ng markers, ng microsatellite markers. Okay? So, parang kinumpare niya lang yung tumor sa normal and hindi nagtutugma yung microsatellites ng tumor sa normal. So, ginagamit tong detection, diagnosis for colon cancer or HNPCC specifically. Okay? So, pwede ka rin gumamit ng ibang microsatellite marker sa iba't ibang diseases. So, for example, natripan mong hanapin yung microsatellite markers for breast cancer. Pwede mo yung gawing thesis. So, sa breast cancer, pwede ka humanap ng microsatellites doon and i-compare mo rin siya sa normal tissue. So, yun actually yung thesis nung isa kong friend. Naghanap siya ng microsatellites sa breast cancer, tapos kinumpare niya sa normal tissue. And in a way, pwede mo nang ma-detect na early kung meron kang breast cancer or wala. So, yun. Maganda siya, yung microsatellites, maganda siya na parang research topic kasi parang general siya in all kinds of cancers. So, or, yun. Okay, okay. So, next disease natin ay xeroderma pigmentosum. And these children are called the children of the moon. And xeroderma pigmentosum is a rare hereditary disease in which children who inherited the disease are very sensitive to the sun's ultraviolet light or very sensitive to the UV radiation. And exposure to kahit few minutes lang in sunlight is enough to cause severe burning of the skin or can cause skin cancer. So yung suot nila, it is a suit designed by NASA to prevent them from the UV radiation of the sun. So this suit, yung suot-suot nila yan, dinesign niya ng NASA para hindi sila magkaroon ng skin cancer or magkaroon ng skin burn. So kaya sila tinawag na children of the moon kasi pwede lang sila lumabas sa gabi or bawal sila lumabas sa umaga kasi nga magkakaroon sila ng skin cancer kapag nainitan sila ng araw. Okay. And xeroderma pigmentosum is ito yung parang mechanism behind xeroderma pigmentosum. UV radiation can cause DNA damage by triggering pyrimidine dimer formation. And accumulation of these dimers will cause cancer. So you have to prevent the accumulation of the dimers by excision repair, right? But in patients with xeroderma pigmentosum, there is a mutation in the genes involved in excision repair. And these genes are XPA to XPG genes. And these genes are responsible for excision repair. So, um, again, in xeroderma pigmentosum, basically, walang kakayahan yung katawan ng patient na magkaroon ng excision repair or ma-exhibit ang excision repair. Kaya yung dimers mo nag-accumulate, therefore causing cancer. And another type of xeroderma pigmentosum is a mutation in the 8th gene called XPV. This produces an alternate form of xeroderma pigmentosum. And what it affects is the DNA polymerase ETA, now responsible for translation synthesis. So, dalawang repair mechanisms yung na-affect sa xeroderma pigmentosum, right? One is yung excision repair, and the other one is your translation synthesis. Because the mutation in your XPV gene, it affects your DNA polymerase ETA. And in xeroderma pigmentosum, you can diagnose it by skin biopsy or genetic testing. So in genetic testing, you can just give a sample of your DNA and para titingnan kung meron kang mutations in the genes involved in xeroderma pigmentosum. So, ayun. And sobrang rare nito, kaya parang hindi pa masyadong develop yung diagnosis for xeroderma pigmentosum. Parang nalalaman na lang na meron kang xeroderma pigmentosum pag lumaki na yung bata. And ito yung itsura ng mga patients na affected ng xeroderma pigmentosum. Okay? The next disease or the next disease in merong relationship sa dysfunction in DNA repair is your breast and ovarian cancer. So, yung BRCA1 and BRCA2 genes mo, they compromise the homologous recombination repair and causes hereditary breast and ovarian cancer. So, pinakita ko lang, di ba, sa homologous recombination repair nyo, sobrang simple. But, there are actually proteins involved in homologous recombination repair. And these are the proteins. So, meron kang ATM protein, merong BRCA1 and BRCA2. And mutations in BRCA1 and in BRCA2 results to breast and ovarian cancer. Kasi itong BRCA1 and BRCA2, they aid in homologous recombination. So what they does is that BRCA1, siya yung protein na mental block ako. So yung protein na nag-i-initiate or parang tinatawag niya yung ibang enzymes to repair your DNA damage here. And your BRCA2 here, it holds a protein called RAD51, which facilitates in yung pagpasok ng ng DNA strand mo doon sa homologous repair. Wait, balikan natin yung homologous DNA repair. Sorry guys, sabog-sabog ako. Okay. So, dito, kapag tinignan mo to ng merong protein, yung, yung mga proteins na yan, right? Yan yung protein. Ito, yung BRCA1, siya yung responsible sa pagpasok nitong damaged DNA doon sa undamaged template. Wait. So, dito mo makikita yung BRCA2. And yung BRCA1, siya yung magtatawag ng ibang enzymes for homologous repair. Diba? Yan yung protein. Balikan natin yung breast cancer. Okay. So, when your genes for BRCA1 and BRCA2 are mutated, ma-affect yung repair mo for homologous recombination. Therefore, your damage will accumulate and causes hereditary breast and ovarian cancer. So, yun lang yung lesson natin for today. So in summary, we discussed about the different types of DNA damage, and we learned the various ways in which DNA repair is conducted for all kinds of damage, such as dimer, single and double-strand DNA breaks. And we also discussed the mechanism behind DNA recombination. And we studied the diseases relating to the dysfunction of DNA repair. So ayun lang, and um, next meeting, we will discuss transcription or from DNA to RNA, and try ko na rin isa ba yung translation. Okay? So, para mas mabilis yung discussion natin. So, oh no, di ko pala to, di ko to napakita. So, diagnosis for breast cancer ay breast exam, and also you can use biopsy and also MRI. So, yun lang naman yung parang diagnosis of breast cancer. So, breast exam, biopsy, needle biopsy, and also MRI. Okay? So, ayun lang. Meron ba kayong tanong?
Ma'am,
Yes po.
ano lang
Yes
ma'am,
po.
na-post na po itong PT na po. Itong PT na po to sa G-Class. Naka-post na po.
Po?
Ito pong PPT, naka-post na po sa G-Class. Ito pong lecture 3.
Opo. Naka-post na po yung lecture 2 sa Google Class. Tapos nakasama rin doon yung link for the recorded lecture. Paki-check po na lang. Meron pa po
Um,
bang tanong? Yes
Yun
po.
pong nucleotide excision repair, saka yung base excision repair, may limit po ba yan kung ilang nucleotides or parehas po ba ng karami ng nucleotides ang parang nare-replace nila?
For base excision repair, parang in base excision repair, specifically na isa lang na nucleotide base pair yung tinatanggal mo. But in nucleotide excision repair, bulk ng DNA mo yung tinatanggal. So, yun yung parang main difference ng dalawang repair mechanism. In base excision repair, isa lang yung tinatanggal niya. But in nucleotide excision repair, meron kang nasasama na neighboring nucleotide doon sa bulk ng merong damage. Meron
So, ma'am, yan pong excision repair, parang form siya ng self-wounding nila.
function ng, sorry, pardon.
Self-mending po. Like, sarili nila, sila din yung nag-aayos.
Mm, inherent to sa cells natin, kaya tayo merong low mutation rate. So, kung walang nucleotide excision repair and base excision repair and all these kinds of repair, homologous recombination and non-homologous recombination, mataas yung magiging mutation rate ng cells natin. But since meron tayong different kinds of repair, yung first na diniscuss natin ay yung DNA polymerase proofreading mechanism, right? Second, yung translation polymerase. Next ay itong mismatch repair. Next ay yung base excision and yung nucleotide sequence. And ito yung sa single strand breaks lang. So for yung sa double strand breaks naman, you repair them by non-homologous end joining and homologous recombination. So the collection of all these types of repair, they happen actually na sabay-sabay sa cells mo. So nangyayari sila actually during mostly in DNA replication. Before your cells, they separate into two daughter cells. In this way, it prevents your DNA na damage na ma-pass on sa daughter cells mo. So these repairs, they happen simultaneously during DNA replication and before the cells separate into two daughter strands para maiwasan na mapasa yung damage doon sa daughter cells. Meron pa
Thank
po
you po.
bang tanong? Okay. May tanong ba kayo dito sa mga sakit? Magdadagdag pa sana ako ng sakit, yung ataxia telangiectasia Basta dito sa ATM protein. Pero, yeah, halos same lang naman yung parang principle. Pag um, meron kang mutation sa gene na involved dito, magkakaroon ka ng sakit. Ay, yung PPT ngayon, lecture 3, hindi pa po nakapost. Hayaan nyo, hindi pa naman ako magpo-post ng quiz para sa lecture 2. And, ano pa ba ang sabihin ko? Ah, 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 ayun lang. Pero, oo nga pala. Bakit ko na nakalimutan? Guys, sabog talaga ako ngayong araw. Sorry talaga. So, balik tayo doon sa umpisa. Hi. Yan. So, dito, yung dapat ko actually nasabihin, sinabi ko ba na yung quizzes nyo, kunyari, magkakaroon tayo ng 10 quizzes for the whole of the semester. Pero 7 lang, yung top 7 lang doon sa scores nyo yung kukunin ko sa grade nyo. So, like, for example, meron tayong, ilang quizzes ba magkakaroon? 15 quizzes. Sabog talaga ako, guys. Kunyari, magkakaroon tayo ng 15 quizzes sa buong semester na to. And sa 15 quizzes na yon, half lang yung kukunin ko, yung top scores nyo lang sa quizzes na yon yung kukunin ko sa grade nyo. So, pwede kayo like maka-perfect ng 7, yun na yung kukunin ko para sa grade nyo. So, 100 na kayo automatically sa quiz nyo. Okay. Bakit ko to ginagawa? Kasi, nagkwento sa akin yung best friend ko na sa med school siya and yun daw yung ginagawa sa kanila sa med school. Parang nakuha ko lang yung suggestion. Tapos parang, okay, gawin ko na rin sa inyo para mabawasan yung stress nyo. Kasi alam ko na stress kayo. Okay? Meron pa ba kayong question? Ano pa ba kung nakalimutan sabihin? Yun, basta tandaan nyo, sa lahat, sa 15 quizzes natin, or 14, half lang doon yung kukunin ko para sa grades nyo. Yung top lang, yung top. So, kung out of 14 quizzes, lahat kayo 10 or 10 yung score mo, edi automatic 100 ka na.
Ma'am, hindi po ba possible na halimbawa yung maling DNA base, andun lang siya sa place na kung nasan siya, pero tuloy pa rin yung replication saka yung transcription?
Yung DNA base, like,
Yung mali po. Like yung
yung
maling, apo.
yun yung sa translation polymerase. So, wait lang, saan na yung slide na yun? Ayan. So, sa translation polymerase, kahit na may mali dito, tinuloy niya pa rin yung pag-synthesize ng DNA. And yung usual na mali dito ay yung thymine dimers mo. So, or yung pyrimidine dimers mo. So, yung kahit na may dimers dito, nag-a-add pa rin siya ng tamang nucleotide bases sa growing strand. Yun ba yung
Siya na,
tanong mo?
siya na po yan, ma'am, yung damage tolerance.
Mm,
Yung sa
yan yung yan yung tinatawag na damage tolerance mo. So, dahil meron kang different mechanisms for DNA repair, pagkakaroon ka ng um, damage tolerance and minsan, kahit na merong damage dito, magkakaroon ka pa rin ng DNA synthesis.
Thank you po, ma'am.
Okay, thank you. Good question. Important actually ang questions nyo kasi nasi-steer ko yung magiging lectures natin based sa questions nyo. So, alam ko na kailangan kong i-focus yung PCR sa laboratory natin dahil maraming nagtanong tungkol doon. Okay? So, important yung questions nyo. So, mayroon pa bang question? Kung wala na, end na natin dito. May question ba dito? Mhm. Kung wala pa kayong naiisip na question, pwede niyo naman itanong sa DNA participation form natin. Okay? So, kung wala nang may questions po ba?
Isa na lang, ma'am.
Okay,
Yun,
go,
ma'am,
go,
sa
go
ano,
lang.
sa Senador mga pigmentosum po ba? Ano po, as in, ma'am, paka-expose, pwedeng skin cancer agad
Hmm.
or like, yun na po yun. As in, ma'am, right after
Yun
ma-expose
yung,
yan.
madali, like, few minutes lang na ma-expose sila sa sun, magkakaroon sila ng skin cancer. So, sad yun, actually. Kasi
Yan
parang
na,
hindi sila nakakapaglaro sa araw.
yan na
Hmm?
yata yun, ma'am, sa ano, sa Midnight Sun
Sa?
movie. Yung sa movie po, Midnight Sun sa Netflix. Parang yun na yata yun.
Ah, hindi ko pa yun napanood. Panoorin ko nga, Midnight Sun. Sige, sulat ko sa watchlist ko. Midnight Sun. Okay, thank you sa recommendation. Meron pa bang question? Meron pa ba kayong question? 39 lang tayo ngayong araw.
Ma'am, yung ganong,
Merong,
yung ganong itsura, ma'am, yung may mga, ano, parang freckles po, yun na po yung normal niyang, ano, or pakatapos, post-exposure na po yun sa sun.
yun yung post-exposure sa sun. Dahil na-expose siya, sobrang common. Actually, yung mata niya, red, red yung mata niya. Tinakpan ko na lang. Yung mga may xeroderma pigmentosum, nagkakaroon sila ng cancer sa skin nila. So, kailangan talaga nila protektahan and post-exposure to sa sun. So, kaya yung NASA nag ano sila, create sila ng suit, protective suit. Mm. Meron pa ba kayong question? Mhm. Sa next lectures tuloy natin, dadagdagan ko yung diagnosis para mas ma ma-appreciate nyo. And kung medtech na kayo, magamit nyo. Kung wala nang tanong, kung wala nang tanong, let's end our lecture here. Sagutan nyo yung forms. And thank you very much for listening to me. Okay, yun lang. Goodbye. Sorry sa sabog-sabog ko.
Thank you
Discussion.
po, ma'am.
Thank you po, ma'am.
Okay, thank you. Goodbye. Ingat
Thank you
kayo.
lang.
Thank you po, ma'am. Thank you, ma'am.
Thank you, ma'am.